All right, hi. So I'm happy to share a project that I've been working on in the Churchill lab on the regulation of protein abundance and genetically diverse mouse populations. There we go. And so when I'm talking about genetically diverse mouse populations, I'm primarily referring to the collaborative cross, which is a recombinant inbred panel, and then their sister population, the outbred DO, diversity outbred population. And I don't need to go into a lot of details with this audience, but these populations are descended from 8 different founder strains, and notably, there's a lot of genetic diversity present across these populations because 3 of the strains were wild-derived. And in terms of, so our lab is collected across really a couple experiments, large quantitative protein profiling data across all 3 of these populations. And you can really view them as representing many causal perturbations in the form of genetic variants that will then potentially have effects on protein levels. And so, just for some context, there's really a similar level of genetic variation present in these populations, really even more compared to many natural human populations, and much more than traditional inbred mouse populations. And, and so there's some nice features, though, from being in an experimental population. Because of the breeding design that was used, there's reduced population structure. There aren't really rare variants to the extent that there are in natural populations, so you have better power to detect to detect genetic effects for the variants you have. And now that we have this data from across all these populations, we can really do some full comparison studies, which is largely what this work represents. And so we've tackled a number of questions here, such as how do sex and genetics affect individual proteins? How conserved are their effects across the populations, which is particularly important because these are really resource populations, right? And but then to go beyond individual proteins, how do these factors affect sort of aggregates of proteins, such as protein complexes, which often have very important biological function? And so now I'll provide a few details about the specific sample populations. For the collaborative cross, we have 116 animals, and that represents 58 strains with a male and a female pair each. For the DO, we have 192 animals and roughly equal numbers of males and females. The DO data actually is an older experiment that was published back in 2016. But for the protein data, this is all liver tissue. We have greater than 6,000 proteins quantified for each population. And this represents multiplex quantitative proteomics from Steve Geege's lab. And it's really top-notch and highly quantitative. I just want to plug the paper that underlies this talk is just out in Cell Genomics. It came out on Monday. There's a lot of more details and stories in there that I can't possibly fit in this talk, but if anything seems interesting, please take a look at that, and I'm happy to talk about it offline too. So I'll start with the quantitative trait, or PQTL mapping that I did in both the CC and the DO. These aren't exceedingly large sample sizes, less than 200 DO and just over 100 CC individuals. So we're primarily powered to detect the cis effects, or I'm gonna call more generally local effects. And so you see that on the diagonal there of both grids. And then the other key thing to notice is that the DO sample is approximately 65% larger. So we're more powered to detect more local and distal PQTL, unsurprisingly. So for comparing the PQTL, and we'll focus on the local PQTL for this talk. First, I'll just look at the overlap in proteins and whether they had a PQTL in the CC or the DO or both, we can see that there's a relatively high overlap, but then we wanted to ask to dive into it a little bit more and ask how consistent are the genetic effects underlying the PQTL, because these represent the same genetic variation. You'd hope that they'd be very similar, and you're also probably looking to sort of replicate findings across the population. And then another question is, do the CC or DO specific local PQTL really reflect specific population specific QTL, or is it more just a difference in the mapping power for a given population? And so we went about this by looking at the founder haplotype effects at the PQTL. And so here's an example for the gene of CYP2D22, which is, so these are, this gene has a PQTL, and on the y-axis is the CC, and on the x-axis is the DO. So, basically animals that have a WSB allele have very high levels of this protein, and animals that have a PWK have very low levels. But you can see that the effects are very consistent. And as a summary, I took the correlation coefficient, and it's almost 1. And so if we look at all the PQTL that were mapped in both populations and look at the correlation coefficient across all of them, you can see that it's just overwhelmingly the effects are very similar between the CC and the DO, which is good. That's what it means. You could have a finding in one population and look in the other population, some previously collected data, and really support and maybe even pull up your belief in that PQTL. And then to get at the mapping question, I looked at PQTL that weren't even fully detected at a relatively rigorous statistical level. And what you can see is that pulls even more of local PQTLs having similar effects across the populations. So it really suggests that the genetic variation have the same similar causal effects in both populations, and really the differences are largely driven by subtle difference in the sample size that affects the mapping power, as well as random differences in allele frequencies. So there's much more in the actual paper, but I'm going to change tracks a little bit and talk about effects on protein complexes. And this was largely motivated by a paper that came out in 2019 that actually used the DO liver data. And what they showed was that how much complexes co-vary, how co-abundant they are, that level of variation in co-abundance has a kind of a wide dynamic range across protein complexes. And the idea being that complexes are somewhat co-regulated. And so we use this, what we're going to call cohesiveness, and that summary is the median pairwise correlation across the protein complex. And here we're comparing that for over 100 annotated protein complexes, and you can see that it's nicely consistent across the CC and DO, though there are some unique differences, and that can kind of allow you to find little stories that then become interesting when you dive into them. And then we wanted to take another step forward and ask more directly, how do genetics contribute to this sort of complex-wide regulation? And so the exosome was an interesting example, which I've highlighted here, where it's highly cohesive in the CC, but relatively uncohesive in the DO. And what we found when we looked deeper was that, so for one complex member, exos 7, there's a local PQTL that's largely driven by the PWK allele, which is often, it's one of those wild-derived strains and often has strong effects. And here I have the CC data shown, split by sexes to show that if the strain has the PWK allele, there's not much of a sex effect, and that PWK allele drives low abundance. then when you look at more members of the complex, we can see that it appears to be then acting distally on other members. And so I've colored these dots by the genotype at exos 7 for the protein exos 3, and we can see that exos 7 locus is strongly controlling exos 3. And so if you expand across the whole complex, you see that same pattern where really having a PWK allele at exos 7 drives just overall low abundance of the exosome. And this is probably driven by some sort of stoichiometric balance that's been observed in protein complexes. And one cool thing is that you can use these data to see effects being exhibited on sort of related but not core members of the protein complex like ETF1 and DIS3L. And so now we wanted to get at why is it different though between the CC and the DO? Why do we not see this strong regulation in the DO? And so here I'm plotting grids of the founder haplotypes at the exos 7 locus that seems to regulate this whole complex in the CC. And so each vertical slice of this grid represents a mouse, and then I have the genotype colored in, technically it's the haplotype dosage. And then the columns are ordered based on the magnitude of the phenotype. So basically this cluster here represents that low PWK effect. But what stands out is if you look at the DO, we don't see that, any sort of low cluster. But if you also look at the bars here, we don't see any individuals that are actually homozygous for the PWK allele.
Greg, sorry, just to note, you're running out of time. You got just
yeah. Okay, thank you.
That would be great, please. Thanks.
Yep. And so, this is consistent with there being a recessive effect in the CC that really gets exposed by the CC being inbred. Conversely, it's dominant in the DO, and you'd really need a rather large DO sample to see all of that. So just for conclusions, I want to say that, we have high conservation of local effects. These populations are good for replicating findings across each other. Recessive dominance can contribute to differences and provide sort of interesting examples. The protein complex co-abundance is well conserved and can even, but sort of contrastingly, can even compound differences then based on recessive dominance, like we saw with the exosome. And please look at my paper for more if any of this is interesting. I didn't talk about sex effects. There's even more sort of interesting stories around protein complexes, and I didn't even talk about the CC strains, how you can use the replicates to sort of identify unique protein patterns as well. And we can also, look at these interactive viewers to explore the data yourself. And here are my acknowledgements for my lab and our collaborators. I really need to mention Qian Zhang. She's my co-first author on this project, and also collaborators at UNC, Marty and Fernando. And I'm happy to take questions.
Yeah, thanks, Greg. We haven't really got much time for questions. Maybe we can sneak in a quick question if anyone's got one. Otherwise, just put them in the chat. Any questions for Greg? No. Okay. Thanks very much, Greg. Again. Okay. Super. In which case we can move on to our next speaker, who is Selcan Aydin, also from the Jackson Laboratory.
Hi, everybody. Let me share things. Is it good? Do I need to swap?
Very good. Thank you. That's great.
Okay. Awesome. Hi, everybody. Today, and first of all, I want to thank Greg for the great introduction that he did. That actually cuts down on my introduction time. Thank you, Greg. I'm going to talk about the genetic dissection of the pluripotent proteome today. And what do I mean by that? So we work with embryonic stem cells, which are pluripotent cells. By that, we mean they can self-renew indefinitely in culture, but they can also differentiate into any cell type. So we can turn them into, you can use them to create your own favorite cell type from them. And this makes them a great resource for just regenerative medicine in general, and also a great model organism to study cell fate commitment. The biggest impediment, though, is the variation that is observed in their handling and culturing, and of course, genetic variation that's observed in human populations. So recently, we actually published, we co-published with from a group in JAX and also our collaborators in the UK, where we were able to show that genetic background does impact phenotypic variability and the stability of pluripotency. So genetic background really determines how likely a cell line is more capable to self-renew or more prone to differentiate. And we were able to map this to a single nucleotide difference upstream of a gene called the LIF receptor. And we're able to show through allele swaps how strong this impact was on the transcriptome and on the phenotype that we were measuring. And also show that this genetic variation does impact lineage commitment and how likely you are to go in which way. So today I'm going to talk about, as Greg nicely put, I'm going to talk about these ES cells that have been derived from DO mice. So DO mice contains this huge genetic variation that lets us identify genetic variants, connect genetic variants to differences in molecular traits. And this is a nice, unique data set where we have matching chromatin accessibility, RNA expression, and protein abundance data sets. So in our previous publication, we mainly focused on the chromatin accessibility and RNA expression data. And today I will hopefully convince you that adding protein on top of these two layers gives us a fuller picture. First of all, the pluripotent proteome itself is highly variable. And we see sex, biological sex of these cell lines as a major driver of this variation. On the left, I'm showing you a principal component analysis, and the first component, which almost explains more than 12% of the variation in the proteome, is a good job in grouping samples by sex. And on the right, I'm showing you some gene set variation analyses where we can connect those sex differences to pathway level, which shows up as females having less enrichment or less activity of genital pluripotency factors, as well as inner cell mass cell proliferation, which is a goal biological process that summarizes one of the important pluripotency properties. And also we see a significant difference in ribosome biogenesis and translation between females and males. We also see females showing a higher activity of Wnt signaling, which is more on the line of pushing these cells towards differentiation. In addition to seeing all of this variation at the proteome level, we also looked at compared the protein abundances to chromatin accessibility and transcript abundance, and saw that there is a lot of co-variation happening. So on the left, I'm showing you the correlation across ataxic peaks, so the accessibility of the chromatin regions and protein abundances. And on the diagonal, we see a lot of high amounts of correlation, meaning that most of the genes are highly correlated to the ataxic peaks. So most of the protein abundances of genes are highly correlated to the ataxic peaks right next to each other, right next to themselves. But we also see these proteins, and exactly there are 37 of them actually, that forms these horizontal bands. So they're highly correlated to more than 100 of ataxic peaks across the genome. And these regions are enriched in genital pluripotency binding sites. And on the right, I'm showing you the relationship on a gene basis between transcript and protein abundance, where we observe a very high agreement for proteins and genes found in the extracellular region or involved in X-linked inheritance or lipid metabolic process. no agreement, no relationship whatsoever in splicing and cytoplasmic translation, and some negative correlation, so opposite trends between transcript and protein in cellular respiration and mitochondrial ribosome. So since we observed this covariation across the 3 layers, instead of analyzing them one by one, we wanted to actually integrate them. So for this purpose, I used multi-omics factor analysis, or MOFA for short, which we can intuitively interpret as a more generalized principal component analysis, where it will collapse all of these many molecular measurements into weight matrices and the effect factor matrices. And so what do I mean by that? So we end up with just 22 factors. So we are summarizing all of this huge data sets, 3 huge data sets, molecular data sets, into just 22 factors. And I think it's kind of easy to appreciate the first one. It shows up is biological sex, as not surprisingly. It explains the first factor explains 5% of the chromatin variation, 10% of the transcript, and 10% of the protein data, and it shows a high correlation to sex. And the other one I just want to point out really quickly is factor 3. So you can see that there's a high correlation to the genotype at the LIFR locus that I mentioned earlier in our introduction. So this is that single SNP that we have identified that impacts LIFR expression and also leads to a transband on chromosome 15, controlling many expression of many other genes. So we do pick that up as a source of common variation across the 3 data sets again. So it's in the chromatin data and the transcript data, and also explains a large amount of variation in the protein data. So usually what we do with QTL mapping is we want to connect this genetic variants to chromatin accessibility as chromatin QTL, or eQTL in RNA expression, or pQTL in protein expression, and then further connect that to a phenotype of interest. So instead, what I did was use these MOFA factors that we ended up with, these 22 MOFA factors, to summarize that common source of variation and map with those. So we can identify variants that are impacting all 3 layers. And hopefully the goal here is that this method will allow us to identify things that we wouldn't otherwise be able to identify in single data sets. They will be below our detection limit, but with this combining of the different sources of variation, we would be able to identify new things. So this is the QTL map I ended up with. And some of these are actually previously identified QTL hotspots that we found in our previous paper. The LIFR, so remember factor 3 was highly correlated to LIFR genotype. So genotype at the LIFR locus, we picked that up. We had found another transband on chromosome 10 that was related to 2C-like state, which I will mention a little bit. And then another one we had previously identified on chromosome 13. But it also lets us identify these new genetic signals as well. Okay, so how does the factor 3 co-mapping on chromosome 15 look like? So on the left, I'm showing you the factor 3 QTL, and this is exactly what we saw with the chromosome 15 transband before. We have this 4-to-4 split, where the wild-derived strains group with NOD and all the other lab strains group on top. And on the right, I'm showing you the new pQTL that co-maps to that region. So these are the pQTLs I could identify just by looking at the protein data that co-map to this chromosome 15 position. But the other thing I can do is pull all of these features that have a high contribution to this factor, all these proteins that I know that contribute to this MOFA factor, and ask what the lot score is at that point, at that position. And hopefully you can appreciate that we are picking up a lot of the pQTLs that we already know, but there are also all these new players that are highly contributing to this factor, which we couldn't have really with significance identified as part of this transband. There's a similar story for the factor 4 QTL. And here we have a wild-derived versus lab strain separation. And again, these are all of the co-mapping expression QTLs in that region. And when I do the same thing and look at all the transcripts that are highly contributing to this factor, we can see that, yes, we are picking up all of these high contributing transcripts that also have strong eQTLs in the region, but there is also this other set that wouldn't have showed up in our eQTL map, but is also highly contributing to this factor. And with that, hopefully I've convinced you that the pluripotent proteome is highly variable, and sex is a major driver of this variation. There is high covariation across the 3 molecular layers, and MOFA enables us to collapse this information into a small number of latent factors. And genetic mapping with those latent factors lets us identify previously known loci and also new loci that we couldn't have picked up before. And I would like to thank everybody. This has been a huge project, as you might imagine. There are so many data sets and so many data analyses. I want to thank everybody, especially Dan Skelly and Steve, who have been great support. And with that, I'll end and take any questions.
That's great. Thank you very much, Selcan, for this. Any questions?
I have one. Could you and perhaps Anne as well comment on differences and similarities between MOFA and the paracliques that we heard about before? As I understand it, from both you can get groups of genes and then eigenvectors of some sort, and then map those. Do you have any opinion?
The main advantage of MOFA, I would say, over paracliques, and Anne can correct me if I'm wrong on this, is it lets you combine things across different scales. So you can include like methylation data, genotype data, so any sort of different types of data sets and combine them together. And I've only used the common cell lines that have all of these 3 data sets in my analysis, but it also lets you do so you can also use kind of an overlapping data sets, say some of them would have only one, so some of them will have only RNA-seq or protein and not the other. So you could also infer missing information through MOFA.
Great, thank you.
Can I ask you whether you could use methylation data within that analysis?
Yes. Actually, their example paper combines RNA-seq data with methylation data, I think, in ESCs.
Great, thank you.
And recently they upgraded to single cell data too. So I think a lot of people use it to integrate single cell measurements.
Okay, can I ask a quick question, if I may? What's your overall hypothesis? What do you think you will find in the end? Do you find regulators that affect maybe the lowest layer and the rest just change by association, or do you find, say, something that sort of overall affects methylation, and therefore you'd expect, of course, a downstream effect, transcriptome, proteome, etc., etc.
I think there is all of them in there because this is such a big data set. So for example, the LIFR story in itself is, we see it in all 3 layers, right? It impacts chromatin accessibility, which then impacts gene expression, which then impacts protein. But there are also cases where it only impacts protein, or there is differences in chromatin and expression, which is being buffered at the protein level. So there's all these different stories in it, I would say.
I guess the overall problem is that the correlation between the transcriptome and the proteome is tenuous at best. You know, we always sort of have this paradigm out there that even if you do it at a single gene level, you know, you find something else picks it up, or there's some other factor that you haven't considered, and it doesn't really translate really well. So finding these overall factors that affect all of the different levels, you know, I wonder whether you might expect them to be quite rare, or do we have already a priori predictions as to what they might be, you know, regulatory genes or something like this along those lines?
Yeah, this data is kind of just cells in culture, so I don't really have any comparison points. Chris did a really great job in explaining how, like, once you have different culture conditions or different phenotypes, I think everything becomes more powerful, and you can kind of do a more hypothesis-driven research. So the way I've been doing things is kind of digging through the data and creating new hypotheses that we can test. And LIFR was the first thing that came up from this data set, which shown to be super powerful in a sense. We were able to kind of dig it down to a single nucleotide that impacted all three layers. In adding the protein data kind of, I think, filters out a lot of the variation that doesn't travel up to the protein layer. But other than that, I think also creates all these new issues that you talked about, right? There's a lot of stuff that's just correlated to each other.
Yeah, okay, lovely. Thanks very much for this, Selcan. We must move on to Jack. Our next speaker is Jack Pattee from the University of Colorado.
Assume everyone can see my screen. Let me know if not. Yeah, nice to meet everybody. My name is Jack. I'm a research associate at the University of Colorado Anschutz. Today, I'm going to talk about work that I've done developing statistical modeling recommendations for single SNP EQTL analyses in the hybrid rat diversity panel. So for people not familiar, the hybrid rat diversity panel is a panel of rat strains that is useful in systems genetic studies, associating genetic variation with genomic features like gene expression, associating genetic and genomic data with higher level phenotypes. And there's some elements of the data that make it relatively difficult or nuanced to model. And so we address that in this project. Again, the goal is to make eventually a statistical modeling recommendation for how best to find single SNP associations with EQTL gene expression phenotypes in the HRDP. To do this, we want to characterize the false positive rate and the power of each of our candidate methods, both in real data and in simulation. Obviously, this is more straightforward to do in simulation, where we actually know what a true positive and a false positive is. We have ways of kind of getting at this in the real data as well that I will discuss. But simulation is clearly an important part of this study because it is definitive in that way. And we want to have our simulated data mirror the real data as closely as possible. So to that end, we assess the heritability, the distribution, and some other factors of the real data and use that to inform our simulation. And we think we come up with a simulation that does actually reflect the true structure of the gene expression data pretty well. This diagram at the bottom shows the three component subpanels of the HRDP. We have two recombinant inbred panels. On the left is the HXB, which is 30 strains. On the right is the FXLE, which is 34 strains. And then we also have 35 classic inbred strains. So these are the four models that we considered. I'll discuss them in order here. First is the ordinary least squares linear model. You think you have a continuous response like gene expression, and you have a single SNP as a predictor. You can estimate an OLSE and test for association of that SNP with the expression. There are some issues with that when you have relatedness among the strains in your panel, as we do in the HRDP. Particularly, this relatedness can induce correlation between the gene expression phenotype for different strains. This is a model violation for the OLSE. So to address that, we use the genome-wide efficient mixed model association, or GEMMA. This is a linear mixed model applied through this GEMMA framework. So this uses a single SNP as a fixed effect that we want to do inference on. The random effects model the contribution of SNPs throughout the rest of the genome on gene expression. And it's important in GEMMA to estimate the covariance matrix of these random effects. And this is generally done via this so-called kinship matrix calculation, and that can be done directly from the genotype data. but there's some nuance around how to do this. One method that's been shown in previous literature to be beneficial is this leave one chromosome out approach. In this approach, you do not use SNPs on the same chromosome where you're testing fixed effects when you calculate the covariance matrix for the random effects. So let's say you're looking at chromosome 1, you're testing fixed effects SNPs on chromosome 1. You don't include any SNPs on chromosome 1 when you calculate the kinship matrix. And so you have 20 different kinship matrix, say, for 20 different chromosomes in the HRDP. The reason that this might be beneficial is that you do not, or rather you guarantee that you don't have correlation between your fixed and your random effects in this GEMMA model because of the principle of independent assortment, right? SNPs on different chromosomes are not correlated. An assumption of the linear mixed model is that fixed and random effects are uncorrelated. So we guarantee that that is the case here. And fourth is that we analyze the recombinant and the inbred panels separately with GEMMA and then meta-analyze them after the fact. This is kind of based on the principle that maybe that these populations are too different to kind of mega-analyze at the same time. You might need to analyze them separately in order to get valid inference. So the data that we have is actually a subset of that HRDP data that I showed on the previous slide. We have gene expression data for the HXB recombinant inbred panel and a subset of 13 inbred strains. So that's 43 strains in total. We have around 13,000 SNPs and around 100,000 gene expression phenotypes, which are measured in brain tissue. So to assess this so-called false positive rate in the real data, which we can't, again, exactly determine because we don't know what a false positive is, but we look at the genomic inflation factor lambda. This lambda is the ratio of the median observed chi-squared statistic to the median chi-squared statistic we would expect to see if there's no association between SNPs and a gene expression. So we get one lambda value per gene expression phenotype. We look at the median of each of the pairwise SNP associations with that gene expression phenotype, and we compare that to what we expect under the null. The reason that we think this is valuable is because we expect, under the principle of sparsity, that the majority of SNPs are not actually associated with any given gene expression phenotype. So the median should be, the median chi-squared statistic that we look at should not be associated either. So the more significant that the median chi-squared observed statistic is, the larger that this ratio goes above 1, and the more pathological inflation we think we might have. The value of 1 is considered perfectly calibrated. So given 100,000 gene expression phenotypes, we assess the distribution of lambda across all 100,000 to characterize the type 1 error of these methods. This plot shows from left to right the distribution of the genomic inflation factor for the linear model, GEMMA, GEMMA loco, and the subpopulation meta-analysis. The takeaway here is that the linear model is very pathologically inflated. The minimum value is around 2, the median's around 3 point something. All the values are so inflated that we basically can't trust any inferential results from the linear model because we have this pathological type 1 error inflation. And that's what we kind of suspected, right, given the model violations of the application of the linear model in this case. So that model is basically out. We won't consider it going forward. Next, we look at the distribution of the genomic inflation factor for the 3 more feasible methods. I've just zoomed in on them here. The best, actually, the median that's closest to 1, which is the optimal value, is for the subpopulation meta-analysis approach. The worst is GEMMA loco, which is around 1.2, and standard GEMMA is in between. But these are not kind of terribly inflated. We think even a value of 1.2 is relatively feasible. So we don't basically exclude any method based on what we see here. This is just further evidence, but it is some evidence that maybe the subpopulation meta-analysis approach is best. So the other side of the coin, we want to see how many associations we can identify with each of these methods. So column 2 here is the number of genes with at least one significantly associated cis SNP. We just looked at cis SNPs here, basically to make it easier on ourselves, I guess. And we considered a multiple testing correction of 0.05 divided by the number of cis SNPs, so a Bonferroni correction. And again, the first row is linear model. We can't really trust these results because, as we saw, they're inflated. But about 30% of genes have at least one significant cis SNP. For the 3 other methods, we see that the most associations are GEMMA loco with 18.7, followed by standard GEMMA at 18%, and lastly is subpopulation meta-analysis with 16.9%. So we see that these proportions are relatively similar, but this actual count in the second column is fairly different. We see a much larger count for the GEMMA methods in the subpopulation meta-analysis. That's because when we apply GEMMA to these subpopulations, we need SNPs to pass quality control for minor allele frequency and for missingness in all of these subpopulations rather than just in the aggregated full population. which is more stringent, which means we have fewer SNPs in our analysis, which means we actually have fewer genes with any cis SNPs, which explains the lower count of genes. This is kind of a pervasive problem that we see in simulation too, and we think is a real issue, is that the quality control is more stringent when you need to do it in each of these subpopulations. This plot shows on the y-axis the negative log 10 of the p-value for the gemma loco compared to the negative log 10 of the p-value for standard gemma on the x-axis. And this is the p-value of the most significant cis SNP for every gene with a significant cis SNP. And this is a diagonal line of slope 1. So points above this line are more significant according to gemma loco than standard gemma. We see that essentially all of the points fall above this line. And we kind of operating under the principle that if a SNP is identified as significant with both of these methods, so all of these black points here, that's evidence that maybe that SNP is actually truly associated. So a method that finds a more significant association is maybe finding better evidence of a real signal. So this might be more evidence that the gemma loco actually does have better power than the standard gemma. So now I'll talk about the simulation study that we did. I'm going to leave out a lot of the details because I don't have time to talk about it. But basically, we want to more rigorously investigate the false positive rate and the power when we actually know the ground truth. So we use real genotype data, real SNP data from the HRDP, and we simulate SNP effects from this point normal distribution. SNPs have a high proportion of having no effect, having effect 0 on the phenotype, but if they are non-zero, they're distributed normally. We look at 3 different simulation settings, one with no cis effect, one with a single cis effect, and one with a random number of cis effects. When we say with a cis effect, I especially mean a cis effect that is disproportionately larger than the trans effects. We often see in EQTL analyses, obviously, that cis effects are stronger than trans effects for the most part, and so we want to reflect that in our simulation as well. So we have 2 goals in this simulation. Firstly, we want to compare the power of the 3 feasible analysis methods, right? All of those 3 excluding the linear model, applied to the 43 strain subset. And then we want to compare the power of the best method for the different subpopulations. So the different subpopulations, again, are those 3 component pieces of the HRDP. And we have genotype data for all of those strains, but not the gene expression data. So we can simulate data for kind of the full HRDP and look at power in the full data. So I'm just going to present results for the single cis effect simulation. The other simulations were similar, so I left them out to save time. So here we have on the y-axis the proportion significant, or essentially the empirical power. The x-axis is the heritability, and then the color of the dots indicates the method, right? So this is the 43 subject subset where we're comparing the different methods. Red is gemma loco, green is standard gemma, and blue is subpopulation meta-analysis. We see basically the superior power pretty apparently of the 2 gemma methods that aren't the meta-analysis, and then in the blue we have clearly inferior power. The red and green points are nearly always overlapping, but we do see several cases where the gemma loco has slightly better power. So that's evidence that the gemma loco probably does actually have a little bit better power than standard gemma. I don't show type 1 error results here, but we analyze those and they all control type 1 error at the nominal rate, given our testing structure with the Bonferroni correction for cis SNPs. So we think that this increase in power is meaningful, and is probably evidence that the gemma loco is the best approach here. And we also note that we have better than 80% power once heritability exceeds about 0.4. Which is pretty good and might inform future study design for power calculation. This is a reasonably good guarantee that we have good power for genes heritable above 0.4.
Jack, you're running out of time. Could you try and wrap up, please? Thank you.
Yes, I apologize for that. Yeah, so I'll briefly go over this. We looked at the 4 different subpopulations. We have really good power for the full 92, which is in the green here. Once we exceed 0.2 heritability, we have greater than 80% power. Similar power for the HXB and the inbred strain, then noticeably worse power for the FXLE. So basically the takeaway is inflated type 1 error for linear model rules it out, worse power for the meta-analysis approach rules it out. Gemma standard and gemma loco are similar enough, but we think that pretty robustly better power, both suggested in real data and in simulation, and no real evidence of type 1 error inflation means gemma loco is best. And then we also we'll hope to inform power calculations for future studies with these simulation studies. And we do have good power in the full HRDP, even with fairly low heritability. So that's it. Thanks, everyone.
Genomics data for 1,000 of them. It's untargeted metabolomics, and we have LC-MS/MS, so we have information that's quasi-quantitative, and also we have information about the identity of the metabolites. And finally, we also have shallow shotgun sequencing data for the same subset of 1,000 rats. So these are all data that I'm hoping to integrate in the near future. But for now, my talk tonight will really focus mostly on integrating host genotypes and cecal microbiomes. And so as I was saying, this is a collaboration with the NIDA Center for GWAS and outbred rats, and also with the Center for Microbiome Innovation, which is based at UCSD. So the data collection was the following. Just a quick reminder, the rats were bred by the amazing Leah Solberg-Wood, originally at Medical College of Wisconsin, and then they were sending siblings to the 3 different centers. So something that is quite important for my studies, that at least in theory, there should be 1 male, 1 female from each SIP ship in each of the 3 phenotyping centers, so Michigan, Buffalo, and Tennessee. And here in Tennessee, we have 2 samples of rats. Some of them were phenotyped for behaviors mostly, but also others were breeders. And so all these rats were, sacrificed and samples taken and sent to the University of California at San Diego to get the genotypes, the microbiome, and the metabolome. So one of the first things that I looked at is whether rats have different gut microbiomes in the different phenotyping centers, and also if they have different microbiomes between the young adolescent rats that were phenotyped in Tennessee and the older adult breeders. And so I used the tool Cheetah to do this. It's a really useful tool that's developed by the lab of Rob Knight. And from this picture here, which is sort of a PCA, we can see, and each color represents a study. We can see that clearly the rats cluster by study in terms of their microbiome. So the rats do have distinct gut microbiomes, depending on whether they were phenotyped in Michigan or in Tennessee or in New York. And there's also a distinction actually between Tennessee breeders and Tennessee phenotyped rats. So my idea here was to initially analyze each of the 4 studies separately, and then leverage the fact that we do have the same population that was sent to the 4 different centers, and that I'm actually using the same pipeline to analyze the data later on. So I'll come back to that. I wanted to go through the strengths of this study, and the first one, as I was alluding to before, is that we don't have siblings in the same cage. And that means that genetic effects are not confounded with cage effects. And we know that cage effects can be quite strong in microbiome studies, whereby rats or mice sharing a cage tend to have more similar microbiomes. So if animals sharing a cage were siblings, you can see that we might misinterpret cage effects as genetic effects. It is not the case here, and the plot I'm showing you here gives you the heritability estimated for a lot of different taxa in Michigan and New York, in a model that accounts for cage effects, and in a model that does not account for cage effects. And you can see there's really very little difference. That's because they are not confounded. And then, as I was saying, also something great that we can do here is to compare across studies. First of all, in each study, Michigan, New York, Tennessee breeders, Tennessee phenotyped, actually not the Tennessee breeders, but the other 3, the sample sizes are large enough to run meaningful genetic analyses. We have at least 1,000 rats in each study, and importantly, the rats come from the same population, and of course the same microbiome and metabolome protocols have been applied, and the same computational pipeline as well. So there's really an opportunity here to compare across the studies. Now, whether we call that trying to see if the results replicate or whether the results generalize is a bit of a question. You know, replication should be the exact same same conditions and animals. It's not the same because different phenotypes were collected in the 3 centers. So I prefer to talk about generalizability, and that's what we want really. We like our results to be generalizable. And something important too is that we have microbiome, but also metabolome data and hundreds of behavioral and non-behavioral phenotypes for each of the rats, or so metabolome for a subset, but phenotypes for each of the rats for which we have microbiome. So there is an opportunity again to put the results that we have for the microbiome in context by comparing, for example, the heritability of the microbiome with the heritability of behavioral and non-behavioral phenotypes. And then, of course, we can do, you know, systems genetics analysis to try to integrate the different levels of data and better understand the biology. And finally, we shouldn't forget this population is highly recombinant, with many recombination events having accumulated over the generations of outbreeding. And so when I studied HS rats for the first time during my PhD already, we had a good chance of identifying, you know, putatively causal genes at QTLs. I estimated that at about 1/10 of the QTLs, we had a good idea of what might be the causal gene. It can only be better now because there's been more generations and more recombination. So we do really have a population that permits high precision mapping. So the first question that I've addressed is the microbiome heritable? And so this is a very important question in its own right, and there's been a lot of debate around this. So for example, I'm showing you here 2 different papers, one from humans that was saying that really there's very little host genetic effects on the microbiome. It's all about the environment. And another paper that came out recently from wild baboons saying all microbial taxa are heritable, and both papers are remarkable. The claims are really well supported by the data and results. So there's my thought about it was that for the microbial traits, just like for other phenotypes, it's likely that there is a genetic basis to them, which can be small, but if we get enough data, if we have enough statistical power, we can detect heritable, well, the heritability, the genetic basis of the trait. So I'll revisit that with the data. Because we can compare across studies, it's interesting to ask whether the taxa heritabilities are correlated in 2 different centers. And also, how does microbial heritability compare to organismal phenotypes? Because we need to remember that heritability is population specific. For example, if you get a population with recombinant inbred strains and you get 100 replicates per strain, you will have a very, very strong heritability, but that, you know, doesn't mean much in itself. I think what's really interesting is to compare with other phenotypes collected in the same population and animals in that case. So first question, is the microbiome heritable? There's multiple ways to sort of address that question. The one that I prefer, I think, is to show QQ plots. So these QQ plots have on one axis, on the x-axis, the expected p-value of the test heritability is equal to 0 or different from 0. And on the y-axis, we have the observed p-values for that same test. And so here, each dot is a microbial taxa. The 4 quadrants here correspond to the 4 different studies. So the null hypothesis, again, is that the microbial taxa are not heritable. And if we see that there's inflation, meaning the dots are above the diagonal, then that indicates that the taxa tend to be heritable. And we can see here that in 3 quadrants, there's clear inflation of the p-values. That is not the case in Tennessee breeders. So there is evidence in those 3, not in this one, but we need to remember that the Tennessee breeders, the sample size is half of the other ones. And so there is no evidence, but it's possible that it's due to a lack of statistical power. So there is evidence the microbiome is heritable across the board of microbial taxa. Now, are the heritabilities correlated across studies? So in each plot here, I have the heritability for microbial taxa that was observed in the two studies. So the first row, we have New York, Michigan here, Tennessee behavior, Tennessee breeders. So for example, if we look at this plot, and I have to say all the axes have the same scale, of course, we tend to see some little correlation in that quadrant here. And indeed, there is a correlation that is significantly different from zero, even after accounting for the 6 tests that we're doing here. So between New York and, well, in New York and Tennessee behavior, the heritabilities tend to be correlated across taxa. So if the taxa is highly heritable in New York, it tends to also be quite highly heritable in Tennessee behavior. But overall, really, there's still little evidence that the heritabilities are correlated. So that could mean that truly there's no correlation or little correlation, or it could be that our estimates are too imprecise and we can't really see the correlation that exists. And so keeping in mind that heritability estimates are population specific, what makes the most sense to me to do is to compare the microbial taxa to organismal phenotypes. And I've split here between behavioral and non-behavioral phenotypes, because we know that behaviors tend to have lower heritabilities than non-behavioral phenotypes. And what we can see on this plot is that the microbial traits, which are in some shade of gray, are indeed have lower heritabilities in HS rats than behavioral and non-behavioral phenotypes. And we do see that behavioral phenotypes, well, so interesting. I'm sorry, I think there might be sort of a swap of labels. I'm pretty sure that the behavioral are less heritable than non-behavioral. So coming back to the 2 papers that were published before, heritability of the microbiome is quite low, but if we have enough data, we can detect it. We can see that it's significant.
Amelie, are you one more minute?
Yeah.
Running a bit out of time.
Yes, I'll wrap up now.
Thank you.
Yeah. So I've been talking about genetic effects across the genome, heritability. Now we are also looking at QTLs and trying to map genetic effects. And something that I'm really interested in doing is identifying QTLs that are observed in all 4 studies, as is the case in this one. And then, of course, we want to integrate the phenotypes and do mediation analysis like Leah presented before. So I definitely need to up my game on that. So I've already acknowledged the NIDA Center, the Center for Microbiome Innovation, and then there's many individuals who are collaborators as part of this. And I just want to say that I'm looking for a postdoc to work with me at the Center for Genomic Regulation on host microbiome interactions, or potentially on indirect genetic effects, which is the other thing I do. So if you might be interested, you can check out my latest paper, and that will give you an idea of what I do on indirect genetic effects. And I'll take questions if we have time.
Thank you. So yeah, my name is Cody Cousineau. I'm a 3rd-year doctoral student at University of Michigan School of Public Health. This is my first CTC, so thank you for the opportunity to present some of my very first genetic work that we've done in the lab. I'm going to be going over how distinct mechanisms underlie cholesterol levels in Chow-fed versus high-fat diet-fed mice. All right, so we know that heart disease is due to about 50% diet or lifestyle and 50% genetics. So we wanted to explore a trait that is complex and driven by both lifestyle and genetics, and so we settled on cholesterol. It's fairly clear that lowering LDL-C results in a proportional reduction in the risk of coronary heart disease, demonstrated in Mendelian randomization studies, prospective cohort studies, and RCTs where statins were administered. The greater the reduction in LDL-C, in general, the greater the reduction in the risk of coronary heart disease. And this especially matters in the context of high-fat diets, such as low carbohydrate or ketogenic diets. So low carb and ketogenic diets are becoming pretty increasingly popular across the US. I'm sure many of you know somebody who is on one or has tried one. And this leads to the importance of understanding how individuals will respond in terms of their cholesterol and therefore subsequent disease risk. Many studies have shown positive benefits of ketogenic diets in terms of weight loss and glycemic control. However, there's been evidence that these diets can increase cholesterol in individuals. So for these diets, a decrease in carbohydrate will generally result in an increase in the fat content of the diet. And with an increased fat content, I'm sure there are many who would be concerned whether this is going to impact their cholesterol levels. So again, understanding the importance that understanding these individual responses is becoming more and more important. So our lab has recently performed a meta-analysis of studies where individuals were placed on a ketogenic diet, as demonstrated here by this forest plot. We can see that cholesterol response varies pretty greatly across individuals. For some people, cholesterol may drop significantly, and for others, it may increase significantly. And this is in the face of weight loss for most of these studies. So in terms of a cholesterol response scenario, there are three that I'd like to bring forward. So in the first scenario, an individual with a higher baseline cholesterol who goes on a high-fat diet may experience a significant increase in their cholesterol after that high-fat diet, demonstrated by the red line. In the second scenario, there may be no increase, demonstrated by the orange line. And in the third scenario, they may experience a decrease after going on a high-fat diet, as demonstrated by the green line. My guess is most people would assume that the first scenario is most likely, but going forward, I'm going to go ahead and argue that this may not necessarily be true. So to start, I'm going to go over some data where mice were used as a tool for exploring cholesterol responses. Mice are great because we can control their diet, their environment, and their genetics, therefore controlling for much of the confounding that occurs in human studies. So for most of the data that I'm going to show, we analyzed public diversity outbred mouse data sets where each mouse studied possesses a unique genotype from each other mouse. Our main question we wanted to explore was, does understanding gene cholesterol on normal chow diet predict high-fat, high-sucrose diet response? So to start, we first analyzed DL mice who were on a normal chow diet. Here's the Manhattan plot that we generated after performing mixed linear modeling. We observed several peaks around chromosomes 6, 11, and 12, and identified 6 SNPs that reached suggestive significance. Of these SNPs, 4 were associated with a general increase in cholesterol, 2 with a general decrease in cholesterol. Our lead SNP on chromosome 11 is displayed here. The graph demonstrates that possessing the TT genotype at this location is associated with an increase in cholesterol on normal chow diet compared to mice with the GG genotype or the TG genotype at this location. And so we next performed a transcriptome-wide analysis study, or a TWAS, on these DO mice on normal chow diet, looking at liver gene expression. And we identified 43 hits that had a significant positive association with cholesterol and 170 significant hits that had a negative association with cholesterol. And here's an example of one of those hits where in both male and female mice, greater expression of FPV1 was associated with a significant decrease in cholesterol compared to mice with a lower expression of FPV1. So now I come back to our original question. Does understanding gene cholesterol on normal chow diet predict high-fat, high-sucrose diet response? So to get at this, we repeated all of the previous analysis looking at DO mice who are now on a high-fat, high-sucrose diet to see if that information we learned from the normal chow diet mice translates over. So right away, when looking at the Manhattan plots, the normal chow one is now on the left, the high-fat is now on the right. We noticed that the plot for the mice on the high-fat, high-sucrose diet had unique peaks. Peaks that were present on the normal chow diet don't appear on the high-fat, high-sucrose diet, and the peaks that are on the high-fat, high-sucrose diet don't appear on the normal chow diet. For the high-fat, high-sucrose diet mice, we found 6 suggestive associations, all leading to an increase in cholesterol levels. Now, when looking at that lead SNP from the normal chow diet mice again, we compared that to the high-fat diet mice, and we see that the lead normal chow diet SNP is inversely related to cholesterol on the high-fat, high-sucrose diet, where now possession of the TT or TG alleles leads to a decrease in cholesterol. When looking at the females on the left columns, we can see that the high-fat, high-sucrose diet leads to an overall increase in cholesterol at all genotypes, but there's a pretty clear decrease in cholesterol in mice possessing the TG and then the TT genotypes compared to the GG. So when we check the TWAS again, we found entirely unique transcript cholesterol associations with no overlap at all. We now observed 69 significant hits with a positive association for cholesterol on high-fat, high-sucrose diet, and 27 significant hits that had a negative association with cholesterol on the high-fat, high-sucrose diet. And again, this is with no overlap between them. And then we can also go back and look at the association of FPV1 expression with cholesterol, where on the normal chow diet on the left, we had a greater expression of FPV1 had a negative association with cholesterol levels. But now on this high-fat, high-sucrose diet, we can see that FPV1 isn't really associated with cholesterol levels in these mice at all. We then decided to generate polygenic risk scores for both normal chow diet and high-fat, high-sucrose diet mice using 3 SNPs. And we can see that the PRS, predicted cholesterol, looks pretty good and lines up well with the observed cholesterol for the normal chow diet mice. But when looking at the high-fat, high-sucrose diet mice, we can see that the correlation is much, much weaker and non-significant. So one limitation that we considered is that each DO mouse is unique and was only placed on either normal chow diet or high-fat, high-sucrose diet. So no mouse received both diets. So therefore, we wanted to know what some of this would look like with genotypically similar mice. And so looking at published data of inbred strains of mice on both normal chow and high-fat, high-sucrose diet, we analyzed their baseline LDL-C, which is on the x-axis, and compared that to their high-fat, high-sucrose-induced LDL-C response, which is on the y-axis, in 2 different studies, both using BXD mice. So based on what we observed, there appears to be no significant correlation between baseline LDL-C and high-fat, high-sucrose-induced increases in LDL-C, arguing that that first cholesterol response scenario that I showed you is not necessarily true. And so then we decided to take this one step further and ask this question of our meta-analysis of human individuals on a ketogenic diet. So far, the data that I've shown you argues that this middle scenario is the truer scenario. So after looking at our available data, we again saw that now here we have baseline LDL-C on the bottom and the change in LDL-C on the y-axis. And we saw that the change in LDL with the higher fat ketogenic diet had no correlation to baseline LDL, again suggesting that the first scenario in terms of cholesterol response shouldn't be just assumed to be correct every time. So to make some brief conclusions, genetic associations for cholesterol with high fat feeding are distinct from those on normal chow diet. Transcriptional associations with cholesterol levels are diet dependent. Baseline LDL-C is not predictive of diet-induced hypercholesterolemia. And we should be thinking in terms of gene-diet interactions and not diet-independent genetic associations. With that, thank you for listening. And again, thank you for the opportunity. I'd like to thank my committee, Dr. Bridges, Dr. Seeley, Dr. Saslow, and Dr. Zhu, and then as well as the rest of the Bridges lab, as well as my funding source. Thanks, everybody. Take questions.
Thanks so much, Cody. That's brilliant. Thank you. Great. Any questions for Cody?
Just to say it's awesome work, and it does call into question the one intervention suits everybody model that we're sort of forced to make very generic recommendations for humans because we can't get accurate data for any one human necessarily. And we see exactly the same thing, Cody, in the BXDs, where we actually also are looking at lifespan, but cholesterol is one of the traits. And I sent you and Dave some data for both chow diet and high fat diet for about 30 strains that will amuse you.
Great. Thank you
It's a
so much for
beautiful
that. Thank you. I really appreciate that. Thank you.
Cody, I liked that a lot, and I might have missed it. Did you talk about the heritability of the traits? Because of course, if the traits had very low heritability, it would be natural that one population wouldn't replicate another population. And it's not because the environmental interaction, it's just because you haven't actually got a very good predictor of it.
Right.
I know your sample sizes are limited, but do you have any heritability or other reason to believe that these things are sufficiently heritable for the expectation that you would see similar in both diets?
Right. I haven't done any heritability calculations yet. That is something that I know we are going to look into, but so far, yeah, we haven't done anything there yet.
So I guess a lot of yours was focused on cholesterol, but do you have some other measures like body weight, for example, which is easy to measure, and I think we have a better appreciation for the heritability of body weight?
Yeah, I mean, we definitely have plenty of data sets with other phenotypes that we can look into. We're pretty interested in cholesterol and how different types of fat or different amounts of fat affect that, which is why we started there. But we have a lot of other data that we can definitely look into.
And you might well have reason to believe that cholesterol is highly heritable, but I just don't know how heritable it would be, and so I don't have an expectation.
Yeah, appreciate that concern. Yep.